As you know that next generation sequencing technologies have revolutionized the pace of discoveries, and same happened as far as those expression studies are concerned. In the previous sections, we have seen that there are large number of issues with microarrays. We have large number of problems where we need to normalize that. It's hard to come up with good slides, and it's hard to come up with the good probes, and then those hybridizations are kind of physical hybridizations, and then the signals which we are getting are actually which are hard to detect them and quantify them in terms of those numbers which we get through the computers. So here is a new technique that is RNA-seek, RNA sequencing. Seek here represents that we get the sequences of the RNA while using those next generation sequencing machines like Illumina or 454, etc. So it's calculating transcript abundance and prevalence by ultra high throughput cDNA sequencing, as Murtazawi et al. Has said it. This was actually the first pioneering paper. Among the pioneering papers, there are two of them actually, they appear together, this Murtazawi et al. And then Wang et al. So where this RNA-seek technique was described in detail. The sequence reads are individually mapped to the source genome, and then they are counted. So we take those sequence reads from those next generation sequence machines, we take the messenger RNA, we convert it into the cDNA, and then this cDNA is then aligned into our reference transcriptome, and then we count how many number of or how many reads they hit to each gene. So that is kind of their relative abundance, and we then get this density of these reads corresponding to RNA from each known exon, splice event. We can capture new candidate genes. So here are the procedures in general. So we have isolation of all messenger RNA. So we get the complete messenger RNA out of the cells, we isolate them. Mostly the extraction protocols, they are using these methods which are capturing those poly A tails. So they are recognizing messenger RNAs by the help of those poly A tails. They in fact design some oligo DTs, so which get attached to those tails and then capture them. And then this messenger RNA, since mRNA is less stable, RNA is overall less stable than DNA, so it's converted into the cDNA using the reverse transcriptase. Then this cDNA is sequenced, and these sequences are then mapped to the genome. So there are different aligners or alignment programs, or we call them as mappers. So different softwares, they do that. The more times a given sequence is detected, most abundantly transcribed it is. So if we have enough sequences, for example, over 40 million, a comprehensive and quantitative view of the entire transcriptome of an organism or tissue can be obtained. So Murtazawi et al. Actually he was working with mouse models. So for animal models, we can say like over 40 million reads. If we get it, we can capture the expression of almost every part of the genome. So overall data analysis is performed in these steps. So we map the reads. Then after the reads are mapped, we can visualize them in genome browser. So where we can see different chromosomal regions and the reads aligning to them. So we can observe some clouds on them. I'll show you in a picture. We can take these reads and put them together. So in this way we can do their what we call it as de novo assembly. So in this way we can capture the overall makeup and an overall transcriptome while using these reads. So we are not aligning with some known transcriptome or genome, but we are trying to assemble the transcriptome with this de novo assembly process. So after you do those alignments, we need to quantify them. We need to count how many those small reads or small chunks or fragments they are there for each gene. So that is called as quantification. And after you do that quantification, we do this differential gene expression. Obviously in quantification, we involve some normalization steps. And finally, when you come up with a list of differentially expressed genes, normally in hundreds, you are interested in finding their functional properties associated with that list of genes. So for that we need to do those functional analysis, or we can call it as a GO term analysis, gene ontology terms. We can put those expression values or numbers of different genes in the shape of different gene networks. So these networks are made by different approaches. So one of them is correlation approach, in which we use the gene-to-gene correlations based upon their, while we comparing their expression profiles, we can fit up a line and we can get their correlation values from that. So here we see the RNAseek in an overview of the RNAseek process. So we have messenger RNAs which are captured with the help of their poly A tails. Then those RNAs are captured that have those poly A. Rest of them, they are not captured. As you see towards the left, there are some flanking fragments. Then these messenger RNAs are converted into the cDNAs. So we see that we have a poly A, and now in the cDNA we have poly Ts. And after that, we prepare libraries. You already know that for NGS machines, you need to size select your fragments. You need to add adapters. So, and then in this way, what we are doing is we are calling in as library preps. So after putting those libraries into the sequencer machine, we come up with those short sequences. So actually, the whole big sequence is broken down into smaller fragments which are sequenced and returned to you as those stretches of ATGCs. We call them as reads. So once you align those reads to a genome or transcriptome, this is an output which we actually see in a format called as SAM or SAM format, sequence alignment format. At the top we have all gene IDs. Actually it was aligned to glyma or soybean. So we have those IDs at the top, and then in the bottom we see one read which is there, starts with this ID, and then we have this glyma 14G07. So that is actually the gene on which this read is mapping. So the gene from which this read is coming. So since these are small fragment reads, we might have those reads mapping to more genes, or maybe since the size is very small, in case of Illumina it's 100 nucleotides, so there can be multiple positions over where these reads they can go and map. So for instance, in this example, we see that this read maps to this particular gene, and then we have some additional flags. We're not going into the detail right now. And we see the actual read sequence, and these sequences are followed up by those qualities. So this is just one read aligning to this particular gene. This format is called as SAM format. So this is a pictorial representation. We have a gene A large number of reads which are attaching to it. The reads are in pointing to one direction. So we have greens and reds. I tell you what greens and reds and blue means. Same way we can have an alternative splice variant of the same gene. So where we can have, we call it as A.1. So we can come up with different transcripts from the same gene. We call them as alternative transcripts. And then those transcripts are put in the databases, and their sequences are there. So since we are aligning with the models, with the gene models and with the all possible transcripts, so we can have a situation like this. We also have a gene B where we have those red dots, and we do have those yellow ones which are pointing on backward direction. So everybody is pointing towards the right, but there are few yellow ones pointing to the left. So this is what is happening is the software first tries to align those reads in normal direction, and then it takes the reverse complement and then try to align that in the opposite strand. So in this way, those are, we call them as reverse reads. So the yellow ones are reverse reads. Green ones are unique reads, which are uniquely mapped, and multiple reads are attaching to, or they can go and align to different genes, and they can align to different alternative splice variants. So remember here, gene A and A.1 are alternative splice variants of gene A, whereas gene B is another gene. It's a different gene. So A and B, there are two genes, and within A we have A and A.1. A multi-read is that can go on to multiple locations, but within different alternative splice variants of the same gene. Whereas a multiple can go anywhere. Same gene, alternative splice variants, different genes. The reverse mapping reads, we have already seen that yellow ones are which not aligning in the forward direction, but the software takes the reverse complement and aligns on that one. So we call it as a reverse mapping read. So reads may be classified based upon their alignment patterns into these 4 types. So after alignment, our next objective is to count how many reads they are hitting to each gene. So in fact, we are doing quantification. For that, we can use our own scripts which we can write, or we can use the known pipelines. So after we do quantify those gene expressions, we can come up with a matrix or an Excel sheet which is like this. So we have transcript A, whose length is 1 kb, and then we have in treatment 1, we observe that there are 23 reads which hit to this gene in treatment 1. In treatment 2, it's 44, and treatment 3, it's 7. So actually, it's basically there are 3 replicates for the same treatment, and we are comparing with the 3 replicates from the wild type. So in this way, we can come up with a count matrix like this. So these are in fact the raw read counts. So in RNAseek, transcript abundance and prevalence is calculated using ultra high throughput cDNA sequencing.